Hi everyone. So today I'm going to talk to you about some work we've been doing to understand different aspects of the global trends in three-dimensional tree architecture. I want to thank my co-authors, but especially want to thank Hank Schugert, which this talk is very strongly based on. So trees come in a huge range of shapes and sizes, ranging from trees with massive crowns to the oldest trees we know of, or trees that look like fractals, or trees that look just strange, or finally, trees that Hank might say are just straight up big as houses. So we can start to look at this range of different tree architectures and really ask ourselves how much carbon these trees have and why that might actually be. And we can't leave our understanding of this down to, I don't know, aliens. What we really need is a plant structural economics spectrum. And this has been proposed a couple of years ago by some colleagues of mine. And we believe that we can take terrestrial laser scanning, this new instrument, and capture these different elements of tree architecture to really better understand why they are the way they are. This here is a fly-through video of terrestrial laser scanning data. And what you can see from this is that there are millions, if not billions, of points in some situations that describe the three-dimensional architecture and capture these elements of structure that we need to understand why trees grow the way they do. And the way we do this is we go into the forest and we place an instrument at multiple different locations. The instrument scans and collects LiDAR distances, that's light detection and ranging, but at billions of times. And then we stitch all these different locations together to give you this really detailed three-dimensional picture that you see here. So we've been doing this in locations across the planet. These are just a few of the field locations that I visited collecting these types of data. But if we plot this across precipitation, temperature, biome envelope map, we can see the range of different environments we're actually capturing with this new database once we've brought in several other members of the community. Now we've already assembled 1,000 terrestrial laser scanning plots, but now we're opening it up to the community, and we believe we could easily have thousands in a huge range of different environments. So that's pretty cool, but what can we actually learn from this? Terrestrial laser scanning really has a potential to revise and improve the ways we're modeling for structure and function. And on the structural side, we can do this with improved estimates of allometric relationships, so relating something simple like the diameter of the tree to the amount of carbon held in that tree, or the amount of volume held in that tree, or where it puts that volume in the tree, the three-dimensional allocation strategy. That information can feed into and improve our remote sensing-based estimates, radiative transfer models, as well as process-based models. On the functional side, we have the potential to vastly improve our capturing of leaf material in the canopy with leaf area index profiles, as well as the leaf angle distribution within the canopy and how this changes at seasonal timescales, all of which can really improve these different aspects of how we're really modeling and capturing forest structure and function. So on the structural side, we can start to look at what terrestrial laser scanning can do. One way we can really improve our structural estimates is through improved understanding of allometric relationships. So the way that we might do that is we take our terrestrial laser scanning data, segment individual trees, and model them in 3 dimensions, capturing the above ground volume of that tree. And so then we can start looking at how the volume of individuals varies with respect to the diameter of that tree and how that changes with respect to the individual species. Like you see on the right here, we've ordered the allometric scaling coefficients according to the individual tree species we're curious about. We can also decompose that even further, as we show in this center pane, into the vertical distribution of that volume and ask how that actually varies with respect to species. And this is what we've done here, basically showing that there is a different structural signature depending on the species that you're interested in the vertical distribution of volume. Now, we can even take this to the root level. Here's an example from Pangara National Park in Gabon, which holds the tallest known mangroves on Earth, reaching around 60 meters in height. If we take these scans from the understory area of this mangrove forest, we see a massive tangle of roots that we can segment and model individually to start to understand what the role and relationship between the diameter of the mangrove tree and the amount of root material that exists within this area in the understory. Also using that to improve our understanding of the distribution or size distribution of roots in that area as well, which could really improve our understanding of flood modeling in mangrove forests. Not to mention below ground biomass estimates, which are extremely hard to understand at this point because often roots are totally obscured. Now, we can take all of these improved allometric relationships, understanding of the root biomass, and use that to improve our estimates of area-wide carbon storage and biomass estimates. And we've done that here, and we're getting estimates around 4 to 5 teragrams of carbon in this 40,000 hectare area in Pangara National Park in Gabon. And as Hank might say, that's a boodle of carbon. But the question really becomes, how close have we been to getting to these really precise estimates that we're getting now with TLS? So we can compare our allometric model we see in red in the top pane here to the Komiyama equations, which are really widely applied in mangrove forests, which we see in blue. And what we think is happening is there's now this systematic overestimate of biomass in these stands because the large trees are basically never captured with destructive sampling. And so if we propagate that to the mapping stage, we can see some significant overestimates just strictly due to differing allometric relationships. Now, I mentioned before that the current global data set that we have of terrestrial laser scanning data covers this huge range of different types of environments. Well, what we can do with that is potentially enable a global analysis of the types of environmental factors driving tree architecture. So if we create a generalized allometric relationship between diameter and volume and then start looking at what controls residual variability or what controls the allometric coefficients of those particular sites that exist in different temperature, precipitation, for example. Here's one thing we find. This is looking at the variation in our allometric scaling coefficient with the change in average annual temperature in a different site. One thing we see is that as we increase temperature, increase average temperature, trees on average get larger. But then this is only to a certain point. Above a certain point, around 20 degrees Celsius, we start to see this decrease in the size of trees at a certain temperature. What's interesting about this is it actually ties in nicely to some of our work from last year, where we were looking at the role of tree height in explaining mortality risk. And we might be seeing kind of another aspect of this really in this type of global relationship. Now we can focus more on the functional elements, or basically the leaves. One thing we can do is we can capture seasonal leaf area profiles. And this is the type of information that could be of great interest to more functional models of photosynthesis. So the way that we do this is we start with a scan of the forest canopy. We separate out the leaves from the wood in that forest canopy, and we use the density of leaves to reconstruct the vertical profile of those leaves. And here we did this at 8 different plots in central Virginia over 35 different time steps throughout the season. And from that, we can start to reconstruct a full forest average profile and the seasonal trend that comes from that. Now, this is a type of information that's not really existed before, and so we're still trying to figure out how to work this in to these more functional models. But we think it's going to be of a lot of interest to the broader community and really improve our estimates of photosynthesis. Another aspect of this is capturing leaf angle distribution within the canopy, because we have these high resolution measurements. We can reconstruct leaf surfaces and give us leaf angle, which we can then also feed into some of these process-based models or radiative transfer models. The way we do this is, again, we separate out the leaf and wood and attach the leaf measurements to those surface angular measurements of the tree of the leaves. Then we normalize according to density within the canopy to make sure that we are correcting for some density effects that we may see. But then we can apply this to any of our sites in the world, from Costa Rica to Gabon. These data sets were collected a week and a half apart. It was a crazy field trip. But this is the type of information that could really substantially improve our estimates on the functional side. So what we've shown is that TLS can really improve these elements and the way that we're capturing forest structure and function by improving our allometric relationships, estimates of 3D allocation strategies that all can feed into these different aspects of either the way that we capture things with sensors, remote sensing, radiative transfer models, or even the dark art of process-based models. And then finally, we can capture functional elements as well that further feed into those improvements. And with that, I'll just say thanks. And thank you to all my co-authors. And if you have any questions, feel free to shoot me an email and follow on Twitter. Also, if you were following along, you might have some key Hank Schugert PowerPoint bingo items to cross off now. And finally, I just want to send a very special thanks to Hank Schugert. You taught me so much in science and taught me just about everything I know about making a good PowerPoint presentation. So thanks so much.